Hello, I'm David Tabb, and today I'd like to talk with you about detecting and localizing post-translational modifications. This is a subject that I just wrote a new talk for because I was visiting the University of Crete at Heraklion, and it's a topic that's been near and dear to my heart for a long time, so I'm glad to get to talk about it in this presentation. I'm going to start with the role of enrichment because there's very few samples that are so dominated by post-translational modifications as they're just released from the cell. We need to talk about how we can enrich for it, create samples that are highly concentrated in modified peptides for a wide variety of circumstances. From there, I'll talk about kind of the oldie but the goodie, the standard way that we go about looking for post-translational modifications using a feature called variable modifications in most database search engines. And from there, we'll start talking about how sure we can be about where a particular modification is located within a peptide sequence. Finally, we'll talk about the use of open modification search. Now, I can show you, I'm standing in front of my slides. You may not be able to read everything at every moment. I would encourage you to use the link in the description for this video on YouTube because you'll be able to download the PDF of these slides and can make notes however you like. Okay, so let's start with the basic premise of shotgun or bottom-up proteomics. We start with a protein mixture. It's been a long time since you've needed to isolate a particular protein for identification and characterization. Today we can do this for thousands of proteins and mixtures. So a protein mixture here may be as complex as a tissue lysate or cell lysate. From there, we're going to start by making the sample even more complex. We're going to use something like trypsin, which is a proteolytic enzyme for digesting up proteins to produce peptides. The peptides are a whole lot more tractable for the kind of an analytical chemistry we're going to do downstream. If you've got a very complex sample, you may need to take multiple shots at it with your mass spec. So it might be that you separate the peptides out to different fractions, each fraction of which is then subjected to LC-MS-MS, which really is represented by all the other icons here. So now that we have our peptide mixtures ready to go into the instrument, we use liquid chromatography, typically C18 beads. So we're separating the peptides on the basis of hydrophobicity over the space of, let's say, an hour. As they leave the column, the peptides are ionized. They're already acidic, but we apply a heavy voltage to them that causes them to create a cloud of positively charged ions entering the instrument. And down here we have two different kinds of measurements that are taking place. We typically have a high-resolution mass spectrum that says essentially which mass-to-charge values have intense signals right now, which is another way of saying which peptides are most concentrated in the sample, in the set of peptides coming off the column right now. So this is going to give us a highly mass-accurate representation of what their masses are, because we can infer charge by things like isotope spacing. And that gives us a lot of information when we try to identify those peptides later. We're going to isolate a whole bunch of ions of the same peptide, maybe 10,000 copies of a particular peptide. We collide those peptide ions with gas, and that collision process produces fragments. They're measured, the fragment ions are measured in a tandem mass spectrum. So we have a mass spectrum saying what the intensity of the precursor was, what its mass was, and then we have a report of which fragments are generated from it. All right, so let's talk about this enrichment step that I was describing a moment ago. If you're dealing with most tissue or cell samples from, say, humans, the greatest likelihood is that the great bulk of the peptides you see are not phosphorylated. So while you can perform a phospho search for phosphorylation sites in a bulk proteome, the reality is that you're going to get a much better sensitivity for these modifications if you are able... Kittens, please don't make noise right now. Thank you. If you're able to affinity enrich for peptides bearing these modifications, you'll have a much better chance of getting an adequate picture of what phosphorylations are present. So the typical way that this is done is the immobilized metal affinity column, or an IMAC. You hear them called IMAC columns a lot, yeah. So IMAC is used then because metal ions have a nice attraction for these phosphorylated peptides. So the metal affinities for iron, gallium, and titanium oxides are all really good for pulling phosphorylated peptides out of a mixture of all peptides. In most cases, what you'll find is that people apply this affinity step after digestion, which means you're enriching for phosphopeptides, not for the proteins in particular that bear them. So it might be the case that you have just one peptide appearing for a particular protein. It needs to be reflected in how you filter the data after the fact. Because if you apply something like a two-peptide rule to say, I only want to see proteins for which I have two peptides in evidence, suddenly proteins that bear one phosphorylation just fall out of your data. Okay, it's worth noting, however, that IMAC is not just good at pulling out phosphopeptides, but also acidic peptides. So if you're trying to look at the data from an IMAC enrichment, you're likely to see a large number of peptides that bear no phosphorylations at all. So we've talked then about the affinity of metal for phosphopeptides, but it might be that you want to do an enrichment that uses antibodies instead. So it's possible to buy commercially antibodies that are raised against, say, phosphotyrosine. And this is a particularly important one if you're focused on the signaling that goes on within cells, because the phosphotyrosine events are important in that process, and yet phosphotyrosine is present in a vanishingly small number of peptides cell-wide. So you have very little chance of seeing lots of phosphotyrosine if you're just looking at gross cell lysates. You got to enrich for that. And monoclonal antibodies are particularly nice for that purpose. You can just buy these commercially. So in this case, in the case of phosphotyrosine, we're playing against two effects. One is that tyrosine is already one of the rarest of amino acids, one of the least represented 3 amino acids, really. And so spotting tyrosine at all is challenging, but detecting it when it's phosphorylated means that we're looking at just a very, very small fraction of all peptides. Antibodies are not the only proteins that can be used for affinity enrichment of modified peptides. And for that, we should really talk about glycans. So glycans are a really special case for post-translational modifications. I'll try to talk through this. First off, of course, we have a variety of sugars that can be attached to proteins. They may be attached in an N-linked way, or they may be attached in an O-linked way. Cats, this really isn't the time to have a loud fight. It's not. So what we see in this case, though, is that individual sugars may just be attached in long chains, or they may be attached in branches. And this is something special about carbohydrates, that these sugars have multiple OHs in place, multiple hydroxyls on them, and each of those may be considered an attachment point. So we can find ourselves encountering branched saccharide chains, and this can give you a very complex structure that you need to look at. So if you were to treat all of this as just one ginormous mass stuck to a particular site in the peptide, much of the mass of the peptide is made up by sugars, not amino acids. Okay, so you'll find that these are attached in vivo within the endoplasmic reticulum of the Golgi process. And then you find that these subunits are able to connect through multiple OH groups, and yet these glycans play a really, really important role in things like cell recognition. When we talk about things like, if we talk about cells that are positive for certain markers, very frequently those markers are glycans. So if you're trying to understand cell recognition or the process of protein trafficking, you definitely need to know something about the glycans that appear in these mixtures. Okay, and one of the valuable things that we can use for pulling out glycosylated peptides are lectins. Now, lectins are not antibodies, but they are proteins that bind to specific carbohydrates. So lectins are to glycans as you would find like antibodies are used for pulling out particular proteins. But it is also possible to find antibodies that have been raised against particular sugar forms of peptidoglycans. Now, earlier I said to you that we mostly use technologies like reverse phase liquid chromatography with C18 beads in columns for separating peptides on the basis of hydrophobicity. However, sometimes when we work with things like glycopeptides, we need to take into account that their chemistry is a little different. It's shifted because of the presence of these glycans. So something like a helic material can help you to separate and retain these peptides better than using something like C18. Okay, so from the informatics side, we're still using a standard database search engine in most cases to do this kind of PTM analysis. There are a wide variety of algorithms designed specifically for PTM identification. In this case, we're going to focus our attention on the workhorse, on database search engines. So we were able to produce the set of proteins possible in this search space by downloading all the proteins for this species. We're able to digest them in silico to say which peptides would result from these protein sequences. And then we have the PTM expander. This is the route that we use for variable modifications. And this PTM expander then bears a lot of weight because it decorates these peptide sequences with allowable PTM motifs. The rest of this is to say we only need to compare those peptide sequences to each MSMS if they have an appropriate mass. We need to have some way of predicting which fragments should occur for a peptide, and then some way to compare the fragments we observed with those that were generated that are predicted for a particular peptide sequence. All right, now I'm going to stand on this side for this. In this case, we have a 9 amino acid peptide. It's one drawn from alpha S1 casein, and it's valuable for making the point of why PTMs expand the search space for proteomic identification. So here we have the bare peptide, no modifications on board. And we're going to pretend we're doing a phosphorylation search, which means if there's a serine or a threonine or a tyrosine, we must consider it in both its base mass and in its plus 80 phosphorylated form. So we have 3 different sites within this peptide that may or may not be modified. You'll see that I've separated this into 4 different colors. Black is the base sequence. These 3 red ones all represent single phosphorylation variants of this peptide. The blues represent double phosphorylation of this peptide. And finally at the bottom we have one that represents all sites modified. So we had 3 sites, each had to be considered in 2 different forms, and that gives us one of the very few equations I'm going to subject you to, 2 possible states, not modified or phosphorylated, 3 different sites that might bear that modification, 2 to the third is 8. So one peptide becomes 8 just by allowing for phosphorylation to take place. Now, this is a little problematic because, as you can tell, it can dramatically expand how many peptide sequence decorations we have to consider in identifying peptides. So if we were to perform a no modification search, this teal ball represents how many comparisons we are making of a sequence to any of the spectra. And as you can see, it's relatively constrained in this space. If we just allow for the single most popular modification, oxidation of methionine, and again, methionine is a very rare amino acid, we see that the search space grows to this magenta size. So we have more sequences that need to be compared to spectra on average, but it's not hugely larger. If, however, we allow both the oxidation of methionine and the phosphorylation of serine, threonine, or tyrosine, we see that the number of comparisons we need to do per spectrum has risen quite substantially. And that's because serine and threonine are relatively frequent amino acids, so considering whether they're modified or unmodified has an outsized impact on the search. So, you might think to yourself, well gosh, I want to search for all the modifications. So if I'm going to consider a dozen different modifications, that would already be very, very aggressive for database search. You simply cannot enter, all the hundreds of different PTMs that are possible into a single database search and get a meaningful result from it, other than a headache and a dead computer. But so if you are going to increase the search space, the chance that a real sequence that is unmodified gets outcompeted by all the peptides added to the search space means that you could lose identifications by considering additional modifications. You could lose sensitivity by doing so. All right, so in this case, we're going to look at how many different modification variants must be compared to spectra. If we have no PTMs, it's a very small number because in this case we're using something like a 10 parts per million precursor mass accuracy. Again, we see that adding the acetylation of the N-terminus or the oxidation of methionines has a relatively small impact. But now, as we add in the possibility that methionine, tryptophan, tyrosine, and proline may gain 16 mass units, well, our search space has grown dramatically. And if we add on top of that double modifications, double oxidations, we see that the number of possibilities has grown and grown. So this, the sample I'm considering here is not a phosphorylation search. None of these were the STY plus 80 mass shifts. This was instead a sample drawn from a 93-year-old human's eye lens. So the proteins and the crystallins in your eye lenses don't really turn over meaningfully. So you get a lot of oxidation events that pile up on these crystalline proteins over the course of 9 decades in exposure to air. So this is a case where the modifications that we see are a dominant part of the sample, not something you have to enrich for at all. Now, a part that really bothers a lot of people is that as soon as you start adding lots of modifications into play, your ability to control the false discovery rate for PSMs, for peptide spectrum matches, has got to be a lot more aggressive. And this is exactly what I was talking about a moment ago, that as you allow peptides to gain PTMs, it's possible that these PTM variants, even when they're wrong, are going to score better than the true sequence with no modifications for this peptide. So in this case, we're dealing with a titanium dioxide peptide enrichment. This is an IMAC column. The data, I think these were from a breast cancer sample, if I remember correctly. We produced 24 raw files, which is to say 24 LC-MS experiments. And then we applied our database search and controlled false discovery rates to limit the PSM false discovery rate to 2%. That's an aggregate false discovery rate, which is to say that it controls the overall error rate. But we can subdivide those peptides into those that have no modifications, that carry 1 phosphorylation, 2 phosphorylations, or 3 phosphorylations. And in each of those pools, we can ask how many of the hits, how many of the PSMs we accepted in this class have come from the decoy search. This is how we control the false discovery rate. And what we see is that the peptides that bear the most phosphorylations have the highest false discovery rate drawn from this. So in aggregate, we controlled our false discovery rate to 2%. But the error rate within the most modified peptides we see is well off the scale. It's much, much higher than 2%. We had almost 20% false discovery rate among the peptides that were identified with 3 phosphorylations. So essentially, the more modifications allowed to be identified on a peptide, the higher the chance that it's a bogus ID. All right, so which modifications actually matter? Do we need to look for everything, or are the great majority of modifications we see coming from a pretty small list? In this case, I have pulled frequencies from the UniProt knowledge base Human Reference Proteome, both reviewed and unreviewed, and I asked which types of mod-res, which types of modified residues have been documented well enough to be stored as part of the human proteome. And what we see at the top is phosphorylation. Does this mean phosphorylation is the most common modification in human proteomes? No. But in terms of those that are documented in UniProt, phosphorylation is number one. Under that, we come to acetylation. There are two ways to think about acetylation. One is that protein N-termini are frequently bearing this plus 42 modification, but the other is that lysines may be modified by acetylation as well. Both of these contribute to the overall frequency of acetylation. In some cases, we see in vivo methylation, most typically on arginine. That adds a plus 14, sort of a carbon and two hydrogens in effect, are the change. We see dimethylation as well. That also occurs on arginine. As you might expect, a dimethylation is twice the mass of single methylation. Succinylation can also happen on lysine. We have a mass shift of 100 on that. And hydroxylation shows up very frequently. And in this case, the hydroxylation is not something we're talking about methionine. That's not an in vivo modification. Methionine oxidation happens because you breathe near your samples, right? And the oxygen in the air is reacting with them. So, excuse me. So hydroxylation of proline is an altogether different thing. Hydroxylation of proline is quite important because in collagen, in solid tissue samples, the collagens have a fair number of prolines, and those prolines are frequently hydroxylated to create hydroxyproline. So that's been documented many times over in the database. There are certainly databases out there to help you learn more about these different modifications. I'm highlighting PSIMod, Unimod, and PTM-list. These have different levels of annotation and kind of different audiences, I suppose. I think of Unimod as being directed very much at the mass spec community, and the resource is drawn from the papers of the mass spec community. PSIMod is an effort to renovate a lot of this PTM information and to make it a little better structured. For example, things like the acetylation of an alanine is handled as different than an acetylation of a glycine. So those are treated as two different mods in the case of PSIMod, but not in Unimod. And then finally we get to PTM-list. Now PTM-list is something you may never have heard of, but I guarantee if you spent any time in UniProt, you've encountered it. UniProt has a controlled vocabulary in which they enumerate all the different post-translational modifications that may be allowed to exist. So if this PTM shows up in the annotation for any protein, it must show up in PTM-list as well. So if you were to look at a particular Unimod entry, you would get a lot of information about that particular PTM. So in this case, I'm looking at trioxidation. I see that its monoisotopic mass is just below 48 mass units. I see that it's been documented in cysteine, tryptophan, tyrosine, and phenylalanine. Helpfully, it also includes information about which papers this information has been drawn from. So you can use this as a way to broaden your reading list and understand better why we believe this is a real modification. This is bringing us to the difficult challenge of localization. How do we determine where a particular modification sits? Now I've brought back our 9-residue peptide that we were looking at a few slides back. In this case, I'm trying to differentiate just 3 variants. All 3 of these have 1 PTM on board. So the 3 of these have the same sequence and they have the same composition of post-translational modifications. Therefore, their mass is identical. In mass spec terms, we would say these are isobaric peptides, identical masses, not close masses, identical masses. The next piece is that is there a very slightly different mass if phosphorylation is connected to a serine than to a threonine? Okay, well, maybe not perfectly identical, but anyway, in this case, we also have the same mass for the modification in each case. Now I would ask you, do these three peptides that differ only in where the phosphorylation is attached, are they going to separate in liquid chromatography using C18 material? The answer is probably not. Are there some slight differences that might creep into their retention times because of this, because of the presence of the PTM at different sites? Maybe. But we would certainly not expect that C18 material will neatly resolve these two separate bumps in separate chromatograms. So when we just see the MS-MS, it's possible that multiple phosphorylation variants exist and all their fragments of all three variants may be mixed in a single MS-MS. But for now, let's pretend that only one of these is the correct answer. How would we deduce the probability that the phosphorylation is at this site and not that site? Okay, so I've colored the fragment ions differently here. So B ions represent the N-terminal fragments from the peptide, and the Y ions represent the fragments that include the C terminus. In this case, we see that B1, B2, and B3 have exactly the same mass as they would if there were no modifications on board at all. But B4, B5, B6, B7, and B8 all contain this phosphorylation. Therefore, their masses are 80 mass units higher than they would be if there were no phosphorylation present. We see the same sort of pattern here. The first 5 fragment ions from the second variant contain no phosphorylation, but the last 3 do. The first 6 fragment ions in the last case do not contain phosphorylation, but the last 2 do. So, if you're trying to decide, is the phosphorylation at this position or this position, you need to ask which of these labels are effectively changing color here. So we see that B4 has changed from contains the mass to not containing the mass of phosphorylation. B5 has changed from containing the mass of phosphorylation to not containing the mass of phosphorylation. So both of these pairs of fragments are shifting in this theoretical spectrum and this theoretical spectrum. Now, if you're trying to make the judgment about is the phosphorylation on this serine or this threonine, there's only one peptide bond's worth of information to help you make that discrimination. That's B6 and Y3. In this case, B6 does contain the phosphorylation. In this case, it does not. In this case, Y3 does not contain the modification mass. In this case, it does. So if you're trying to evaluate, does this spectrum represent this peptide or this peptide, you have a really big challenge because only 2 fragment ions could possibly tell you whether it's here or there. And it's quite frequently the case that these fragments just go missing. So if it's absent from the MS-MS, you have no ability to discern whether it's this peptide or that one. This is the challenge of localization. So this peptide with this PTM explains the spectrum, but do we have the position of that PTM correct? Very frequently, if you're making a big announcement about some PTM that you've discovered in a Nature Biotech paper or something, you're going to need to justify that this modification is the only real explanation, and to be really clear about just how much information you have for localizing it to this position. It might be that multiple PTM positions create a tie for this particular spectrum. And it may be that in some cases an individual MS-MS represents data from fragments of multiple modification variants of that peptide. So systems like A-score have been written to rescore, and effectively, what this peptide spectrum would look like if we move the modification mass from AA1 to AA2 to AA3 to AA4. At each of these places we can rescore this PSM, and it gives us some notion of how much better the position we've chosen is than any other in the spectrum. There are some approaches that make use of original search scores, not just of the best match to this spectrum, but of the second best and the third best. If, for example, you have very little scoring difference between the best match for the spectrum and the second best, it may be some indication that there's greater ambiguity here than you would like. And down at the bottom here, I've listed 4 papers. Again, you should be able to read those more easily in the PDF if you download it, that have been used to produce these scoring systems for different localizations of PTMs. All right, now I need to talk about open searching, because open modification searching has become a really common way that people go about exploring their data, asking which mass shifts are most dominant in this set. You've seen this diagram before. The PTM expander stuff is still in place, and yet, in this case, we see that the peptide mass filter has been replaced. Now we're saying any decorated peptide within 1,000 Daltons, that's just an arbitrary number, that any decorated peptide can be compared to more or less any MSMS. If we can find similarities, it might be that we have a mass shift of unknown mass that is here to represent this MSMS. So one of the things that we frequently see from this are mass shift tables. So up at the top, we have columns that represent each of the amino acids, and down the rows, we have different unit masses to say, for example, methylation would be about 14 Daltons, so information about potential methylations would appear on this row, 14. Now, what we're looking at is kind of a very broad look at a bunch of data from formalin-fixed, paraffin-embedded tissues. These have been problematic because they bear a lot of modifications structurally. The way that the samples were pickled or preserved has created modifications to the proteins in bulk. So in this case, we're using a kind of a color strategy to flag us to which parts of this table are most important. When you see these red boxes, these represent the largest numbers of counts. The counts here are how many peptide spectrum matches have been found to add an arbitrary mass to this particular amino acid. So when we see something like this red box for methionine with 3,339 PSMs in it, that's the highest value on this chart. And if we look over the row, we see that 16 is showing up. So in this case, the oxidation of methionine is the most common mass shift seen in all of these data. Not surprising, because formalin-fixed, paraffin-embedded samples are in contact with the atmosphere, and even before they come to your lab, they already bear a large amount of oxidation in them. All right. Now, if we look down the row here, we see that proline is also showing up with 1,289 PSMs. And if we chase that over to the side, we see 16 is showing up there as well. So methionine oxidation is dominant, but so is proline oxidation. Remember that collagen, that protein I was talking about quite recently, is a major component of extracellular matrix. Samples from formalin-fixed and paraffin-embedded tissue have a lot of ECM within them. Therefore, they're going to have a lot of collagen and a lot of hydroxyproline. But these tables are a way to reason out, to summarize all the different mass shifts, understanding which amino acids are most likely to bear them. It's worth mentioning that modifications of peptides and of proteins are just one way that we can change the mass. It's also the case that many of the amino acid changes have similar masses to PTMs. So if we consider the case of glycine and alanine, glycine has a mass of 57, alanine has a mass of 71. The difference between them is CH2, a methylation change. So if you see a plus 14, maybe it represents a methylation of lysine, or maybe it represents that a glycine in the sequence database, maybe the wild type sequence, has been replaced by alanine in this particular person's tissues. It certainly can happen. It might be that just an oxygen is all that separates the masses of two amino acids. There's also this kind of happy family here that we see aspartate and glutamate differ by only a methyl, and asparagine and glutamine differ by only a methyl, and in turn, aspartate and their corresponding asparagine and glutamine differ by adding an NH and subtracting an oxygen. So this family of four is very tightly related. So there, just for your information, remember that the mass shifts we see when doing PTM searching may not be from modification at all. They may come from sequence variants instead. And a really important thing to talk about is that boring is more often correct than is brilliant. If you see a plus 22 mass shift on a peptide, it probably means there's a little bit of sodium left in your sample. It's not been perfectly desalted, because 22 is what happens when sodium is creating the ion of this peptide rather than a proton. If you were thinking about this in terms of amino acids, you might think, oh, this aspartate has been replaced by histidine. It's the same mass shift, but the probability that you had some salt left in your sample is probably a much safer explanation than something like there was a mutation. Blind PTM searches, things like the open modification search that we talked about, they can reveal patterns of mass shifts, but you should be really careful about trusting any of these PSMs that result. Use open modification searches to guide you to the best possible standard database search. And in general, those standard database searches are going to control false discoveries much more effectively. Keep in mind also that there may be something other than trypsin causing digestion points within your proteome. If that's the case, it might be that what looks like a negative mass shift, a loss of mass on the peptide, is what's happening because you have a semi-tryptic peptide that doesn't make it all the way to the next trypsin cutting point because another mechanism led to the cleavage of peptide bonds. Always be careful of the warp, spindle, fold, and mutilate problem. If your algorithm can do basically anything to its mass ladders, this excessive search space is going to do a lot to promote false matching. And a lot of the PSMs you result in will be erroneous, even if you try to control for false discoveries as much as possible. Essentially, the more degrees of freedom that you allow your search engine, the greater the proportion of false discoveries will be in the modified peptides. Similarly, then when you look at the PSMs that result from these searches, if a single PSM bears lots and lots of post-translational modifications, we should have a lot of doubts about them. They're quite frequently false positives. So PTM detection has been part of MSMS identification from the beginning. I put a little star on this in my mind because the Sequest publication was 1994. It wasn't until 1995 that the dynamic or the variable modifications were added. But this variable modification approach comes at some cost. It's not just a slower search, it's often a less sensitive search as we add more and more PTMs to the mix. Certainly paying close attention to false discovery rate control is pretty important. The localization is going to be quite necessary for you to introduce a new PTM that you've detected in key proteins. So be ready to document the evidence that allows you to localize it here rather than there. certainly interpreting the results of open modification searches is going to require a lot of detective work. You need to think about all the different possible reasons that this mass shift might be seen in this peptide. Cultivating your skepticism to avoid making any statements that are not absolutely justifiable given your data is quite essential. These things can help to make sure that your result is publishable, defensible, and robust against the future. So hopefully all of this has given you a lot of ideas about how to go about hunting PTMs in your own data. If you'd like, you can drop me email. My email address is down here at the bottom of the slide. If you make a comment on this YouTube video, I'll try to catch it, but I often forget to check YouTube messages. So it might be a month before I spot it. If you make a comment on the video, feel free to send me an email. I'll try to respond to it when I can find the time. But thank you for your attention, and I hope you found this video useful. This channel has loads and loads of different videos on proteomics, so feel free to use all of these however you like. And hopefully I'll be recording another video for you soon. Thank you very much.